Buenas tardes. Bienvenidos a este seminario del programa de Frontiers in Genomics que organiza el Centro de Ciencias Genómicas, la Licenciatura de Ciencias Genómicas y el Instituto de Biotecnología. Y como siempre, en estos seminarios, las preguntas las pueden hacer los que están aquí presentes al final, alzando la mano, y los que están conectados por internet pueden subir las preguntas al chat, y ya ahora lo que va a pasar es que van a aparecer en la pantalla para que sea más dinámico el asunto. Bueno, habiendo dicho eso, para mí es un gusto presentar a la doctora Qian Peng, que es profesora asociada en el Departamento de Neurociencias en el Scripps Research Institute. La doctora Peng hizo estudios de licenciatura en Ciencias de la Computación en la Universidad de Beijing. Después hizo una maestría en Ingeniería Biomédica en la Universidad de Tennessee y su doctorado en Ciencias de la Computación en la Universidad de California en San Diego. Desde 2012 al 2013 hizo un postdoctorado en el Scripps Translational Science Institute. Interesantemente, también después del 2013 al 2014, realizó otro postdoctorado en Biología Humana en el Craig Venter Institute. Desde 2015 al 2016 fue postdoc en el Departamento de Molecular and Cellular Neurosciences, nuevamente en el Scripps Translational Science Institute. Desde 2016 al 2022 fue Assistant Professor en el Departamento de Neurociencias en el Scripps Translational Science Institute también, y a partir del 2022 es Associate Professor en el mismo departamento. Qian ha recibido reconocimientos muy importantes, y solo para mencionar algunos, recibió el NIDA Avenir Award in Genetics and Epigenetics of Systems Abuse, el NIDA NICE Early Career Investigation Showcase Award, the Enoch Gordis Research Recognition Award de la Research Society of Alkalines, un fellowship del NSF. También pertenece a sociedades importantes como la American Society of Human Genetics, the Research Society of Alcoholism, the International Society for Biomedical Research on Alcoholism, and the International Society for Computational Biology. Y el día de hoy, Jen nos va a hablar de Genetic Architecture of Addictive Disorders. Thank you, Jen, to be here. It's a pleasure, and we're really excited about your field of research, and we're ready to hear your talk.
Thanks for the invitation, and thanks, Mario, for the introduction. Good afternoon, everyone. So I'm going to tell you some of our research on genetic architectures of addictive disorders. So first, I'm going to describe some background research and the findings with regard to the variations of alcohol and substance use disorders across populations or ethnic groups. And then I'm going to tell you some additional approaches we use to study the addictive disorders of the genetics of the addiction. This includes ancestry-informed analysis, rare variant-based analysis, and the Platropic detection. So first, some background. Harmful use of alcohol and drug use have been a worldwide public health issues. About 6% of all deaths are contributed to alcohol each year globally. This rate is even higher for people aged between 15 to 49. It's like 10% of the deaths are attributed to alcohol. So in fact, alcohol is the leading risk factors for people of this age group for the premature mortality and disability, etc. And about 5% of the global burden of disease and injury is contributed by alcohol. And hundreds of millions of people are disabled, have health issues, have other disease, are all due to the alcohol drinking. Similarly, drug use disorders, this including illicit drugs and nowadays some legal drugs, also have a very major impact on human health globally. About 500,000 people die each year due to drug use, and 35 million people suffer drug use disorders, hundreds of million people have actually used the drugs, and some of them might eventually become drug dependent. Even though alcohol use disorders are in general pretty prevalent around the world, but this prevalence of the disorder also varies very significantly across different populations and ethnic groups. So the figure here shows is resulted from epidemiological studies in the US population. So what you can see is on the, sorry for that. Okay, so on the y-axis is the percentage of the population that have alcohol use disorder, and the x-axis it just indicates each ethnic groups. These are again in the US population. So as you can see, both Asian American and African American have relatively lower rate of alcohol use disorders, while American Indian really has a very high level of alcohol addictions. And this is, so let's see, okay. So the fact that Asian and African Americans have a lower rate of alcohol use disorders are partially, somehow blocking thing, I think, are partially attributed to them having some of these protective genetic factors in the alcohol metabolic genes. All right, and similarly, other substance use disorders also have a very, their prevalence also very significantly across different populations and ethnic groups. So this figure is just an example for the cannabis use disorders. Again, you can see this is American Indians has the highest level of cannabis use disorders, especially in the severe cases. Again, these are US data. All right, so this actually, in fact, substance use or addiction is really among the most urgent health problems in the American Indians in the US, right? So we wonder what may have contributed to these variations and also to addiction in general, right? So both alcohol and drug use disorders have significant genetic components as well as environmental components. So taking the two most used psychotropic drugs, substance, alcohol is the most used. The second is cannabis, for example. So Qian and family studies have attributed between half of the variability of alcohol use disorders and between 50 to 70% of the cannabis use disorders variabilities to genetic variations. But if you use the entire genome to estimate how much of the phenotypic variation can be contributed to common genomic variants across the whole genome, this variability becomes between 5 to 10% for alcohol use disorders and around 10% for cannabis use disorders. These are accounting for hundreds of thousands and even millions of common genomic variants. And of which the largest genome-wide GWAS study, genome-wide association studies, have only found 10 loci for alcohol use disorders and 5 for cannabis use disorders. So this can really only explain a tiny portion of the heritability. So this is so-called missing heritability problem. And even more so, all these largest results, okay, all these results from the largest GWAS studies, the findings are really from primarily European descent, right? So we have two distinct but related questions here. One is, where are the missing genetic factors underlying addictive disorders, such as alcohol disorders and cannabis use disorders? And the other is, what are the distinct genetic factors that account for population variations? And in order to begin answering these questions, we need to look beyond GWAS, that is genome-wide association studies, even though we know GWAS is really a very important tool and has produced many important findings. And we also need to look at some understudied populations, especially those high-risk populations, because they may have genetic variants that contribute to the disease differentially through perhaps convergent pathways or alternative pathways. So additionally, we found that severe symptoms of disorder are even more heritable than the general average diagnosis. So this may enable us to detect even more genetic factors. So of course, in order to study all this poses certain challenges. Most of the GWAS studies are based on array-based genotyping. So these are, you take a set of variants across the genome, even millions, but still mostly common variants, which means are usually about 5% of the allele frequency. But this is not covering the entire genome. And in order to look beyond GWAS, in order to look at some of the variants of lower frequency, we need population sequencing, such as whole genome sequencing or exome sequencing. And some of the understudied population are a mixed population. Thus, we have to be sensitive to ancestry. Excuse me. And while we have to control for the the population structures in all of our studies, we should also take advantage of the ancestral informations in our studies. However, a lot of the time, some of the tools, the existing tools, are not really made for studying mixed populations. So you also need to think about developing new computational approaches. And of course, we always have to be sensitive to statistical power in all of our analysis. So we took the following approaches. We had 3 high-risk cohorts of 3 different populations. These again are all American populations. American Indian cohort, this is from California, and Mexican American, also from Southern California, and the European American, which is nationwide, but they were more recruited because they have higher level of alcohol use disorders and some other substance use disorders. And all these 3 populations have high levels of alcohol use disorders and cannabis use disorders, and some also have other drug use disorders. So this will enable us to study the severity of these drug use disorders. It will really give us more power to do this kind of research. And all of these individuals are comprehensively phenotyped, in addition to making a diagnosis. There are basically what we call instrument is a whole book of interview questions. So every one of them will be evaluated. They will spend half day in the lab, and there are technicians who will go through all the questions with them. And there will also be some measurements such as EEG and all that. And then, of course, afterwards, all these phenotypes, all these interview questions will be entered into the database, will be analyzed, and the phenotypes will be generated from them. So this gives us the ability to study more complex traits, and will also allow us to derive quantitative severity phenotypes from drug use disorder, what we call clinical course events. So this again will increase power for our association studies. And then we have whole genome sequence, and for one of the populations, the exome sequencing from this cohort, which allows us to study the influence of the ancestry on addiction, as well as to look into the rare or low frequency variants and to look at how they might impact all these addiction phenotypes. And additionally, I'm also going to talk about some Platropic analysis we carried out using a public database and plus some machine learning techniques. So first, let's look at some of our ancestry-based analysis. I showed you earlier that American Indians in the US population have the highest rates of alcohol use disorder. There has long been the belief that American Indian metabolize alcohol differently. And alcohol is primarily metabolized in liver and liver and upper digestive tract by alcohol dehydrogenase. This is this path, and aldehyde dehydrogenase. This is this path. And so these two enzymes, there are many forms of these enzymes, may affect the risk of development of alcohol use disorders because the variants can influence how we respond to alcohol. And the polymorphisms in these genes vary significantly in different populations, because of the ancestry and mixture, etc. And for instance, so these three particular variants, two of them are on ADH1B. This is one of the alcohol dehydrogenase gene, and the other, there's another variant on aldehyde dehydrogenase gene. So these three known variants are highly protective against alcohol addiction. And what you can see here of this variant, there's 70% East Asian have this variant, whereas only 3% of European descent have this variant. While another protective variant for East Asian is ALDHT22. 17% of East Asian population have this variant, was pretty much absent from all other populations. And the African has another protective variants. They have about 20% of the allele frequency, was pretty much zero in East Asian and European. So in this particular study, we wonder what the relations between alcohol use disorder and these genetic variants in this alcohol metabolic genes in the American Indian cohort. And then we also want to determine whether there's an influence of the American Indian ancestry on the relation between alcohol use disorder and these genes. So American Indians are an admixed population. And again, I'm talking about American Indians in the US, but some of them could also apply to American Indians in other parts of America. And the admixture was through intermarriage between ancestral American Indian and European. Admixture sometimes can introduce risk or protective genetic variants. And many generations later, some segment of the genome may be more American Indian or more European. And so a variant may be associated with American Indian ancestry or European ancestry. And the allele frequencies of these variants also vary. So, well, as I said, we need to control these ancestry structures as confounding factors in all of our studies. We really also want to ask the question, how do ancestry differentially influence complex disease such as alcohol use disorders in ethnic groups? So when it creates problem for us in certain studies, we need to control them as a confounding factor. But on the other hand, we want to actually study them to ask the question, to interrogate the influence of the ancestry. So to study, to answer this question regarding ancestry, we really first need to determine the ancestral component of each individual in a population. So here we estimated ancestral makeup of our American Indian cohort. using the variants across the whole genome, right? So each vertical line here, of course, it's not clear. You can't quite see the line anymore. Each vertical line represents one individual in the AI, in this AI population. And the y-axis is just the percentage of the ancestry. And it's sorted by the person's Native American Indian ancestry component. And the red, of course, is American Indian. The blue is European. A little bit of yellow is from East Asian, and the green is from African. So you can see this estimation used in the genome pretty much confirms that this population is admixed between American Indian and European. And of course, this admixture varies quite a bit between individuals. And I should mention that these American Indian cohorts were living in rural areas, but in reserve the areas. And this continues the reserve area, even though they belong to different tribes. So you can think of the environment are kind of similar. And none of them is living in the urban area. And next, we took the genomic variants in the alcohol dehydrogenase gene regions. There are 7 of them. And then we conducted association analysis using 2 alcohol use disorder phenotypes. So we used the variance component model to control for both population structure and some relatedness in our data. We found quite a bit of a significant variance. Of course, many of them are in linkage, right? And some are, some are positively associated with with these 2 traits. These are considered potential risk factors, and some are negatively associated with traits, which are considered potentially protective, right? This is similar to a Manhattan plot, except it's just a regional plot. So the y-axis indicates significance. The higher the number, the more significant they are. And I should mention that in this particular cohort, we didn't find any protective variants from those 3 variants I mentioned earlier that are protective in East Asian and African. And then we examined the effects of ancestry. So not only we can estimate the ancestry from the entire genome, we can also estimate the ancestral component of a small chromosome segment of the genome. That way we can determine the possible ancestral origin of that part of the genome. So I refer this as local ancestry as opposed to the global ancestry. So this graph shows y-axis is the local American Indian ancestry around the ADH region, the region we studied, versus the global American Indian ancestry. Just to show they are, of course, you expect them to be correlated, but they're not exactly the same. And then we stratified the samples by the degrees of their American Indian ancestry and the level of their phenotype. So this particular phenotype is called alcohol-dependent drinking symptoms. The higher the number indicates the more severe the alcohol use disorder. So the individuals in the first quartile of the local American Indian ancestry around ADH are considered of having low American Indian degree, while individuals in the fourth quartile of this local AI ancestry are considered of having high AI degree. So these are blue color. So if you look at the trend here, you can see that the higher degree of AI ancestry, the blue ones, are associated with more severe use disorder. all right? But then, what about the relations between American ancestry and the variants we found? So the figure on the right shows the allele frequency of the risk variants. These are those blue colored variants. Shows the allele frequency of these variants in the two subsample groups. One group are the peoples who have low American Indian degree. These are the red colors. And the other group is those having high AI degree. And each line here represents a variant. And as I said, the y-axis is just allele frequency plotted against alcohol dependence. And similarly, the bottom figure shows you these allele frequencies of the potentially protective variant. So what you can see here is the higher degree of American Indian ancestry is associated with a higher prevalence of this potential risk variant, and also associated with the lower prevalence of the potentially protective variants. So this is pretty much a conclusion of this study. In the next study, we were motivated by evolution-based hypothesis regarding addiction. So one theoretical consumption concerning the native people is that isolation and endogamy over many generations, coupled with a long history of dependence on foraging and subsistence agriculture, may lead to selective enrichment of traits that increase food consumption when highly calorie food is available. This is so-called Swiftogen hypothesis. Basically, you try to preserve fat as much as possible. You also try to consume more when things become available. And the same selective pressure may have caused the enrichment for genetic variants that influence the risk for consumption of other high saline substances, such as alcohol. So this is the hypothesis. And many Aboriginal peoples around the world with a history of isolation, such as Australian Aboriginals, Maori in New Zealand, Pacific Islanders, North and South American Indians, they all have highly increased risk for substance use disorders. And American Indians share certain genetic characteristics with other isolated native people. So this may give us some clues to understanding the genetic underlying the alcohol use disorders in this type of population. So one such genetic characteristic is in this population that are somewhat isolated, this is the enrichment of the what we call rounds of homozygosity. So what is the rounds of homozygosity? This is a long stretches of homozygosity. They arise in an individual when individual inherits two copies of the same haplotype segments from each parent. This, in fact, happens, is basically all around in our genome of every one of us, so they're everywhere. However, the distribution of these ROHs reflect the process of population size reduction, consanguinity, that's like inbreeding. In many cultures, there are cousin marriage, so those could also show certain patterns of ROH, and also admixture between populations and natural selection. And the population isolates tend to have higher burden of ROH, and of course, enrichment of long ROH. This is sometimes due to consanguinity. And ROH has been associated with many diseases and traits. This is because recent inbreeding can enable rare deleterious variants to occur as homozygous. And also reason inbreeding tend to generate long ROH. So this is often the motivation for studying ROH in disorders. But genomic regions under selective pressure may also exhibit characteristics of ROH. This is because selection purges deleterious variants or elevates haplotypes frequency around a favorite allele. However, what benefited the population in the past could contribute to disease risk in the present day disorders, right? So there are certain characteristics of the ROH if they were under selective pressure, because these ROH, they survived many generations. They tend to be shorter when compared to ROHs resulted from recent inbreeding. So our question here is, are there areas of genome with ROH that might harbor genes that confer risk for alcohol use disorders in American Indians? So first, of course, we have to basically to detect all the ROHs in each individual. This is just to show you an example, the distribution of the ROH in this cohort. And what I show here is on chromosome 1. The y-axis is individual, x-axis, of course, is just a genomic position. So you can see these are pretty typical of ROH distributions. These long horizontal lines, these are long ROH, and some vertical lines, those are ROHs that are shared by multiple individuals. Then we can basically compute ROH abundance or ROH burden for each individual. That's basically the total amount of ROH relative to a person, relative to the whole genome, or the total number of ROH. Or we can also determine what we call ROH island or ROH pool. This refers to the regions where multiple people share segments of ROH. And then we're going to study these regions. We're going to study the ROH burden of individuals. And we also computed the American Indian ancestry components of each of these ROH elements. So first, let's look at the relationship between ROH abundance and American Indian ancestry. Each dot is a person. Y-axis is this person's ROH, how much of this person's genome is in ROH regions. And the x-axis is American Indian ancestry. So basically, this ROH abundance first decrease as an individual's degree of American Indian goes from 0 to about, I think here was 23%, and then it increases with the AI ancestry of this individual in a quadratic fashion. We could fit a quadratic curve here. So this relationship is expected because American Indians are admixture of the population, and usually people, if someone is very admixed, you tend to have a smaller amount of ROH. So we totally expected this kind of relationship. And then I didn't have the data to show here, but we also found that the genomic regions in the ROH have a significantly elevated AI ancestry compared to the rest of the genome. So next, we actually did the association study. We basically look at the association between ROH abundance and the severe the severity level of alcohol use disorder in this cohort. Basically, what we found is that the higher the ROH abundance, the more severe AOD is in an individual. We used multiple models, basically considering different sets of covariates, and they all show the same results. And then we stratified ROH by their length. And then we want to see which length group of ROH are contributing to this association. And we found that basically it's the moderate sized ROH between 1 to 2 million length. This is the group that dominated the association between ROH and the AUD severity. So this is kind of consistent with the selective pressure hypothesis. That doesn't mean it confirms it, it's just the result consistent with that hypothesis. And next, we scanned across the genome and did association analysis for each of the ROH pool with the AUD severity. Then we found 2 significant ROH pools. The top one is on the chromosome 1, this red island. This is quite common. This exists about 18% of the individual in this cohort have this ROH. And the total span of this ROH consists 44 genes, but the core region has this gene OSBPL9, which is associated with lipid metabolism, oxidative stress, and inflammatory response. And when we did the functional analysis on the 44 genes in the entire span, we found some enriched traits, including hippocampal tail volume and CSF alpha beta 42. These are all associated with cognitive functions. We also found some enriched immune signatures, including toll-like receptor 3 and 4 regulation, TGF beta 1 targets. And the other enriched functions we found is adipogenesis. And also we look at the CAS scores of all these variants within this region. CAS score basically indicates how deleterious these mutants are. The higher the value, the more likely these variants are deleterious. And over 100 of these SNPs have CAS score of 15. That represents about 3% of the most deleterious variants. And our second ROH that is associated with AUD severity is a rare ROH pool. I think only 5 individuals have this ROH pool, and it contains a total of 9 genes, but the core gene is an axon guidance receptor. Again, 60 SNPs in this region have CAS score over 15. And since there is really no public available replication for this AI population with both the type of phenotype we study and the genome sequencing, so we really cannot replicate our study in the same type of population. So what we did is we just searched for all the GWAS results in the genomic regions of the top ROHs for AUD or any related phenotypes in whatever populations. So we found quite a bit significant variants associated with different phenotypes, alcohol use disorder, dependence, drinks per week, etc., in different populations, including European, Japanese, Han Chinese, Australian, and Hispanic. So to summarize this part, we found that increased genetic liability and lack of protection for alcohol use disorders for American Indian in major alcohol metabolic gene regions relative to other ancestral groups. Through ancestry and admixture analysis, we found that the higher the degree of AI ancestry, the higher the prevalence of potential risk variants for AUD traits in alcohol metabolic gene regions. And we also found that increased ROH are likely risk factors for severe AUD in this American Indian cohort. And it's the moderate size ROH that dominated this relation. So likely resulted from inbreeding of distant common ancestors when the population experienced the size reduction and the subsequent bottleneck and admixture. So the ROH island, we found that associated with this severity contains genes involved in lipid metabolism, oxidative stress, and inflammatory responses. All these are consistent with the Swiftogen hypothesis. So next, let's look at our rare variant analysis. So one theory of the addiction says that not only addiction involves positive reinforcement associated with the rewarding effects of the drugs or alcohol, but it also involves negative reinforcements. That is, you drink to relieve pain, to relieve negative affective symptoms. So these are usually associated with severe use disorders. And these are what we call the dark side of addiction. And so in order to study this severe type of use disorders, we studied three phenotypes. One is a severity measure of the overall alcohol use disorder. This is basically a weighted occurrence of alcohol-related clinical course life events that gives us quantitative traits, which will increase power for association studies. And then we'll also study the two dark side symptoms. One is basically to ask, did you ever experience feeling anxious or depressed if you stopped or cut down drinking? Basically, this is a withdrawal symptom, feeling anxious during withdrawal. The other question is, did you ever feel depressed more than 24 hours during drinking? So this is what we call depression symptoms while drinking. So these are two dark side symptoms, plus these both are binary traits, and plus a quantitative trait that is indexing the overall AUD severity. So we took a rare and low frequency variant approach. Basically, we take the variants that at most with a 5% minor allele frequency. We did a gene-based analysis for two cohorts for both American, formed the two sets of variant group. One group includes rare and low frequency variants on exon and promoter regions. And the other, this is exon plus what we call regulatory promoter regions. And the other group only includes non-synonymous variants and the splicing site variants. And then we just did the association study using the grouped rare variant genes. We did a burden test. Then after correction for multiple tests, we found 3 genes that are significantly associated with severe AUD. 2 of them are for American Indian cohort, 1 for European American cohort. And KCAK2 gene also found nominal significance in the other cohort, in the European American. Similarly, the PDE4C that is significant for European American also found the nominal significance in the American Indian cohort. And what are these 3 genes? So KCAK2 is a potassium channel gene. This is the gene that is associated with severe alcohol use disorder in the American Indian cohort. This gene plays a key role in the cellular mechanism of neuroprotection, anesthesia, and it's a pan-sensing gene. And the second gene, PD4C, is a pro-inflammatory mediator gene. This gene is, we found it to be associated with severe AUD in the European American cohort. We know that PDE4 inhibitors have long been recognized as anti-inflammatory agents, and these inhibitors reduce alcohol consumption and preference in animal models. And the third gene we found that is associated with severe AUD in the American Indian cohort is EBI3. This is a subunit of cytokine. Again, this is involved in immune response. Next, we applied rare variant analysis to cannabis use disorders. But this time we did both a gene-based test. We also did a pathway-based test. So what we did is we downloaded about almost 3,000 pathway gene sets from a public database. Then for each cohort, at this time, we did the test for all three cohorts, American, Indian, Mexican American, and European American. And for each pathway and each cohort, we again formed the two sets of variant groups. So just like before, one group only contains the exome and the promoter variants. The other contains only the basically the subset of the previous one contains only the non-synonymous and the splicing site variant. So we did find some significant pathways associated with COD severity. So one pathway, we actually didn't find any pathway for the American Indian cohort after multiple testing correction, only one suggestive significant pathway. And this pathway that is suggestively associated with the COD severity in American Indian is related to very long chain fatty acid metabolism, which is relevant here. And but we did find the three pathways that are significantly associated with COD severity for Mexican American. The top one is associated with post-translational protein modification. And the second pathway is related to ECM, actual cellular matrix organization. And the third one is really, it's a disease, it's inflammatory and autoimmune condition. And we did find the one pathway that is significant for European American. This is a glycogen metabolic pathway. And this is just to visualize the top 10 pathways associated with COD severity in each cohort and the shape, you can't quite see. So this is American Indian, Mexican American. The third column is European American. The shade of the cell indicates how significant this pathway is. The little star indicates these are the four significant pathways, and the circle just means they're nominally significant. So basically, you can see there are, for instance, the top pathways we found for European American is unique to this population. It's not significant for any other two population. While as the top pathways we found for Mexican American also found some significance in the Native American. And this is actually among the top 10 pathways we found for the American Indians. But in general, if you look at the top pathways, you can see most of the pathways are distinct to the particular cohort. While we identify some distinct pathways for each cohort, we also want to know how much the top pathways overlap between cohorts, right? Because a pathway contains multiple genes, so we can really examine how many genes overlap between the pathways. We're not limited to say if the same pathway is significant for each cohort. So what this graph illustrates is the similarities between top pathways identified for each population cohort. So each node or vertex represents a pathway. The color corresponds to a particular cohort. Red is American Indian, green is Mexican American, blue is European American. And the size of the circle corresponds to the significance of this pathway. Whilst the edge, the width of the edge indicates how much of these two pathways overlap. We basically computed based on the percentage of the genes overlapping between these two pathways. And the cutoff point we said is at least 10% of the gene overlapping. So essentially, we find several clusters of pathways that they share certain genes, and these are also shared between our cohorts. So again, because each node is a pathway, what you are seeing here is really a network of pathways. So just eyeballing it, you can see that there are overlap and shared genetic factors between different cohorts and different populations, essentially. So to summarize this part through rare variant analysis, we identified new loci for AUD severity with converging evidence from both American Indian and European American. There are two genes here, NK2 and the PD4C. And we also identified a new loci for alcohol-induced affective symptoms specific to American Indians. I didn't talk about one gene that is specific to European American. And through a rare variant pathway-based analysis, we identified new pathways for COD severity in Mexican American and European American. And we think we found that these three populations, American Indian, Mexican American, and European American, likely have both shared and distinct pathways associated with CUD, cannabis use disorder severity. So last part, let's look at some pletropia detection analysis we did. So alcohol use disorders and other substance use disorders, we know they're complex disease. They're also comorbid with many other diseases. So take for alcohol, for example, alcohol use disorder oftentimes comorbid with other substance use disorders, for instance, cannabis, nicotine, and other stimulants such as cocaine and meth. And so one question one may ask is there any common process of addiction? In addition, substance use disorders are often comorbid with other mental illness. And for alcohol disorders, it can also be comorbid with liver disease and other metabolic disorders. So another question we may ask is, are there shared genetic factors and underlying a substance use disorder and other disease? So this is the study of pletropy. Pletropy is when one gene affects multiple phenotypes or multiple disorders. And sometimes a pletropy can be expected. For instance, this particular gene, interleukin 23 receptor, is associated with two inflammatory bowel disease. So that's kind of expected pletropy. But sometimes the pletropy can be unexpected. For instance, this gene LRK2 is associated with both Crohn's disease and Parkinson's disease. Basically, one is inflammatory disease, the other is a neurological disease. But nowadays, of course, we know even neurological disease have inflammatory components, so it's not really that unexpected anymore. And another concept is when multiple genes affect one trait or one disease, these are called polygenicity. I mean, essentially, all this disease we're studying here, all the addiction disorders are polygenic. That's what we call complex disease. In reality, what we really have are both polytropy and polygenicity. So the algorithm I'm going to describe quickly is we essentially modeling polytropy and polygenic simultaneously. So what we did is essentially we took all the variants right now, about 200,000 from the entire GWAS catalog. Depending on when you downloaded the catalog, this number keeps increasing. And we intersect them with the genomic variants in each of our cohorts to result a set of variants in all cohorts that also exists in the GWAS catalog. And then we use a machine learning technique to select a best set of variants in each of our cohort for the particular phenotype we're studying. And this we refer as Platropic variants. Basically, it's a variant selection problem. Then we ask the question, using this select set of variants for the disease in each cohort, we ask the question, what other disease and traits or a disease category are enriched in this variants, using the GWAS catalog to answer this question. And I think, yeah, presently there are about 2,500 traits in the GWAS catalog. Again, these are increasing. So we applied this scheme in two studies. First, we applied this method to multiple substance use disorders in American Indian cohort. We look at 4 types of substance use disorders. Alcohol, these are cocaine stimulants, marijuana or cannabis, and the multi-substance use disorder, MSUD. That basically is if somebody is using multiple substances either at the same time or one after another throughout their lives. And all the phenotypes are highly correlated. So we ran through our method and we selected over 200 SNPs for the alcohol use disorders. And then we did an enrichment analysis on disease categories. So what we found, So what we found, the most enriched category is inflammatory measurement. But in fact, the most significantly enriched group is neurological disorder. The stars indicates significance, while the length of the bar indicates basically the odds ratio or the enrichment ratio. And here I just basically listed all the traits underlying these 3 categories. So you can see under inflammatory measurement, we have, like interleukin measurements and interleukin receptor antagonist measurements, etc. And under a neurological disorder, of course, you expect some alcohol dependence traits will show up, but there are a whole set of other psychological disease or mental illness will also show up in this list. And then, well, actually other traits, the group is basically not well grouped, that this is a huge group, but these are the traits that are selected for the SNP. There are some of the traits are kind of unexpected. For instance, this cleft palate and all this. So we consider these variants to be really just potentially pleiotropy, and pleiotropy could also be through linkage or through mediated pleiotropy. And so one question is really, how does the gene affect multiple seemingly unrelated traits. So there are multiple possibilities. I'm not going to, I mean, we didn't really do the studies on the possibilities, but one could hypothesize what could be the reason underlying this. One possibility is one gene product affecting various cells. The other is in the cascading signaling pathway. This pathway is affecting multiple targets. And of course, the other could be a developmental effect. And for instance, like inflammatory ECM, that's an extracellular matrix. This can affect alcohol disorders, but they can also affect many other things. For instance, there's a cleft lip, this kind of trait. But again, this is just for hypothesizing. And then for the other use disorders, and for the substance, the multi-substance use disorder, we again selected over 200 traits. The most enriched disease category is again inflammatory measurement. And for marijuana use disorders, the most enriched is neurological disorder. Yes. And then for the stimulant, cocaine stimulant use disorders, the most enriched, this liver enzyme measurement looks enriched, but it's not significant. The most enriched is immune system disorder, followed by neurological disorder, and also, I think, hematological disorder. And then if we just do a Venn diagram to see, whether these select variants overlap, we found indeed that there are both shared and distinct select loci for 4 of these substance use disorders. And then there are 2 of these variants are selected by all 4 use disorders, and they correspond to 2 genes. One of them, the gene CSMD1, has already been associated with cannabis dependence, schizophrenia, and cocaine phenotype, etc. So we kind of ask, perhaps these are, we can call them addiction genes. And then just based on what number of distinct SNPs versus the number of SNPs that are selected by multiple substance use disorders, we found that marijuana actually appears to be the most distinct. It has the highest number of distinct SNPs being selected, or and the fewer overlapping SNPs. And then we did some functional analysis on this on the genes that are associated with the SNPs that we selected. We found that pretty much it captured functional networks underlying addiction, including neurotropic factors, immune responses, extracellular matrix, and cell adhesion molecules, and circadian regulations that is related to sleep. And then we also did some tissue-specific gene expression enrichment analysis, and found that these pleiotropic genes are differentially expressed in many tissues, brain tissue involved in addiction, such as the top one, substantia nigra. In the next study, we applied our pleiotropy analysis method to cannabis use disorder severity in all 3 cohorts this time: American Indian, Mexican American, and European American. We're trying to find perhaps maybe there are genetic factors shared by different populations. What we found is that for American Indian, we select 105 SNPs, and in this first, okay, so the length of bar is the enrichment, and the number here is the number of SNPs selected corresponding to this particular trait. So we found that in the American Indian, Alzheimer was the most enriched disease, with 4 variants selected. And also, there's the PHF-tau measurement, which is a biomarker for Alzheimer's disease is also enriched. There are 5 variants associated for this particular trait. And what most enriched for Mexican American is prostate cancer, while for European American is a brain volume measurement followed by intelligence. And the body measurements are enriched for both Mexican American and European American body weight and the BMI adjusted with hip ratio. And then we look at the pleiotropic genes that are selected for COD severity in more than one cohort. And we look at, how many of them, what they are. And indeed, there are a few. For instance, a protein was selected by both Mexican American and European American, even though the exact variants differ. This gene is also associated with schizoaffective disorder, even though it's not reported in the GWAS catalog, but it's reported in literatures. And similarly, 2 genes related to calcium-dependent cell adhesion were selected by both American Indian and European American. An afferent receptor gene was selected by all 3 cohorts, and the associated traits are either Alzheimer or biomarker measurement for Alzheimer. So this gene is also implicated in the developmental events, especially in the nervous system. So to summarize this part, we developed method to detect potential pleiotropy for admixed population using a machine learning technique while leveraging large variant trait association database. And we use this method to study the common substrates of addiction and identify genetic factors shared by different populations for COD severity. So our study suggests pleiotropy between substance use disorders and many other complex disorders, essentially. So what have you told you so far? So basically, we have found that alcohol use disorders have polygenic inheritance and are associated with certain chromosome regions and genetic variants within those regions. We've identified multiple new genes associated with AUD and related phenotypes, and also certain genomic regions. And we think potentially elevated genetic risk factors might partially explain higher rates of severe alcohol use disorder in the American Indian cohort. And we found that American Indian, Mexican American, and European American populations likely have both shared and distinct pathways underlying cannabis use disorders. With respect to the computational approaches we took, we found that ancestry-informed approach might give us insights into the origins of the genetic regions harboring risk and the protective variants in different ancestral populations. Using a rare and low frequency variant, both rare and low frequency variants might be a potential source of heritability for addiction. And pleiotropy detection leveraging large public database can lead to new understandings of addiction and may give us new hypotheses for further research. So that concludes my talk. I want to thank all our research participants, without whom, of course, all this work would be impossible. And of course, I would like to thank all my colleagues. This research is really built upon decades of work by many people. And last but not least, all this research are funded by National Institute on Drug Abuse and National Institute on Alcohol Abuse and Alcoholism from the US government. So with that, I thank you. I can take questions.
Thank you very much, Qian, for a very interesting talk. So now the seminar is open for questions. There is some. Yeah.
Very interesting. As you mentioned, it's not surprised, but I'm still surprised that immune regulation and inflammation is playing a major role, at least by your genetic analysis, in addiction. So, but have you looked more carefully and see whether it's a pro-inflammatory response or more like a response that is trying to avoid inflammation and therefore affecting all these recompense circuits in the brain?
Right. So I didn't look at them personally, but these are thought of more of a cycling effect, or they feed back in each other. So of course, because I mostly studied alcohol and other addictions too, but alcohol obviously itself can cause a lot of inflammation of the organ. So and then so it's not surprising to actually find genes that are also associated with inflammation, because this inflammation itself can further your addiction. So this is like a cycling method. It's more like a feedback system. But I didn't look at the neurocircuitries myself.
And the other question is regarding because it's a complex process to become addicted to a substance. So in the case of the protective variants that you found in the alcohol processing, those protective variants have more activity and therefore you have less levels of alcohol in the system, or how does it work?
Oh, so for those particular variants, so okay, let me see if I can find them. So for the, okay, I can't find, but anyway, so there are, remember this alcohol metabolism has two stages. First is alcohol is metabolized into aldehyde. This is using the ADH genes, the protein product of ADH genes. And then the second stage is from aldehyde to acetyl. This is using the aldehyde dehydrogenase genes. So the mutations on the ADH genes, they speed up this process from alcohol to acetyl, because acetyl is the real toxic chemical. And the mutation on the ALDH2 slows down the process from from aldehyde to acetyl, slows down the second stage, which further accumulates this aldehyde in your body. I think I might have said it wrong. If it's an aldehyde that is toxic. So it's an aldehyde, this intermediate chemical that is causing like hangover, causing a flushing effect, causing you feeling nauseous and heart rate going up. So this is the one that is making you very discomfort. So basically these two variants, so if one has the first two variants on the ADH, then you will have a very quick conversion so that you have a quick accumulation of aldehyde in your body. But if one also has the second variant on the ALDH, then you can't metabolize the aldehyde very fast in your body. That also causes even more accumulation of aldehyde in your body. So this together, I mean, having just one of them is enough. If you have both, you might not be able to drink at all if you have both. Some East Asians do have both. So yeah, so this is basically through a very adverse effect. It's like you feel very, very uncomfortable.
Questions? There is one question in YouTube from Fabel Lopez.
Okay. Oh wait. Oh, the last one, right?
Yeah,
that's the only thing I can read. Could the development of AUD be coupled with polygenomic analysis of variants of ADH or the other? Be coupled with polygenomic analysis, phylogenomic. Yeah, I would think so. I haven't done any of that, but that would be very interesting because ADH, ALDH, or in general, these alcohol metabolic genes exist in primates, in other mammals, also in flies. So in many branches of these animals, because we need these genes, basically all animals need these genes to deal with fermented food, right? Primates will eat fruits, and fruits often are fermented. So they have to have a way to deal with alcohol. And I think there was some indication that the early primates, they can deal with alcohol, but they cannot metabolize as much as alcohol as we do. So there was some hypothesis. One of the genes, I forgot the ADH4 or 5, there was some mutation happened on the early primate that made that gene very much more active in terms of metabolizing alcohol, which allowed humans to drink rather than just to eat fermented food. So I would think it'll be interesting to study the phylogenetics of that. So, but also this region, this ADH region on the chromosome 4 is very complex. So people have spent a lot of time on just studying the region because there are a very complex linkage. There is difficult to to separate exactly which variants are doing what. So it will be interesting to study the phylogenetic genomics of that. And the second question is, how would the pipeline work for less admixed population? Pipeline of, actually, if the population is less admixed, it's easier. And if you do GWAS or any association, but nowadays, we always try to control a bit of a population structure. So because even though a population may thought to be not admixed, for instance, European population, there are still population structures in the population. I mean, different Europeans have some different variations. So the usual practice is you always control for some PC principal components of the population structure. But you don't have to use more complex method to control for that. If you don't have admixed population, if you want to study ancestry, then you just have to study different population, which is actually easier. I mean, if you study European, you study East Asian, then you just compare the variants. I mean, in fact, this ADH genes I showed you, even though the rate, the allele frequency is very high in East Asian, you still have about 3% in the European population. And these genes have really been consistently found in any of these association studies, in any, because most of studies are in European populations, but so it will also, it will be found in the study of in the European population, even though the allele frequency is low, because the effect of that mutant is very strong. But of course, in order for it to become very significant in the European population, you need a larger sample size. Whereas if you study the East Asians, you study Chinese or Japanese, you really don't need that many many individuals. I think a couple thousand will already give you very significant findings in the genome-wide association analysis. I think there's a recent paper on that. So if your population is now admixed, it's simpler. Yeah.
Okay. I don't see more questions in the chat.
Yeah.
Okay.
Well, thank you for that interesting talk. And I was wondering, you talked about some variants that I think they're fixated, where I mean, the mutation rate is low, I would think. But what happens with genes that are responsible for neurological disease or disorders, that once the person is under that disease, it will develop the like how to explain the sort of chemical environment for other mutations to develop that you could correlate with this abuse disorders. I don't know if I.
Well, so okay, so this for other mutations to develop within a human, these are the concepts usually is more like a cancer genomics, because here what we study are not somatic mutations, germline mutation, and your chemical environment can cause a lot of chemicals change in your body, but it does not necessarily change your germline. It doesn't change your germline mutation, but it could change. I mean, yeah, I mean, if you have cancer, of course, it will cause that. So usually we don't quite look at them in that light, but the chemicals could change other things other than DNA. It could change gene expression. It could change methylation. It could change other epigenetic signatures. So those people are looking into. And we are also trying to get some data to study. Yes, those are definitely can impact the development of the disease and also the progression.
Thank you.
So I have a question. So I have been thinking about genetic mechanisms that could be involved in the generation of addiction. And for instance, I wonder if there are already experiments in which model organisms like mice or rat has been artificially induced to be addicted to alcohol. And then if this animal has a progeny, what happened with the progeny? If the progeny is more sensitive to be addicted than initially the mother was, and then what happened with the next and the next generation? You are inducing for several times the addiction to alcohol, because if this something is transmitted epigenetically at the epigenetic level, maybe that is the way to know if this may happen, could happen.
Right. So, I myself don't do any animal studies, but animal studies in addiction is a huge area, because in fact, a lot of the understanding about neurocircuitry of the addiction came from animal studies, because it's difficult to study human brain. And so yeah, there are animal models that are addicted to alcohol. Mice are usually not that much, but rat, certain line of rat mutants, these are, through certain labs that they select and they have their own mutants. This rat love alcohol, so they can really be addicted to alcohol. Then of course all their progenitors are, because it's a mutant, so the line will be addicted to alcohol. So they will study their behaviors.
Genetic mechanism, not
Right,
an
right.
epigenetic one.
Right. There's okay. So for the epigenetics, right now we're actually looking at we got some data, but we're in the process of looking at them. It's like you put on some rats. These are more or less regular rats, and you give them alcohol vapor, and then for a certain number of days. You can do it during the development, or you can do it while they're adult. And then you sacrifice them, you took the brain tissue. Then yeah, we do both transcriptional analysis and target-seq analysis to try to see what's epigenetic markers. We do see a difference between the control groups and the vapor groups. And we haven't looked at any methylations, but I know there are groups who have looked at them. Now, obviously, both people and the rat who have been drinking, there are methylation changes on the DNA. So yes, so those, and whether this can be further inherited, I would think it can be, because epigenetic markers can be inherited for a couple generations. Right? It might disappear like a couple more, but usually one or two, some are inherited. Yeah. Okay.
Thank you. And just to finish, so I mean, a very interesting question of Fabel about phylogenomic analysis. I wonder if it's known what kind of variants have the Neanderthals in the ADH, ALDH genes. The Neanderthal organ, the Neanderthals.
Oh, Neanderthal.
That's what kind of variants they have in ADH and ALDH.
That's interesting. Yeah, I haven't looked at them, but I'm pretty sure they have them. They have to have the genes.
Yeah,
and,
of course.
But I don't know what variants. Yeah, actually, I will look at it. That's
Okay,
that's a very interesting question.
I think that there are going to be more Europeans as well. I
Yeah,
think.
they are more European.
Okay. Well, thank you very much.
Thank you.
Yeah, thank you, Qian.
Thanks,
Very interesting, and thank you all of you.
Thanks for being here.
Next week we are not going to have a seminar on frontiers, but the next after that we are going to start. Okay. Gracias a todos. Nos vemos.
Thanks.